domoic acid, it acts on our nervous system. So, but it's a different types of action. The mechanism of action is an agonist to our glutamate receptor. They compete with glutamate in the brain for the receptor. And what happens is that it will trigger cell degeneration. That means cell damage. So when this toxin reach the brain, it damages the hippocampus. So this is what happened on the mechanism of action. So what DA do? So they also induce this neurological and gastrointestinal symptom after ingesting the contaminated shellfish. So this include like nausea, vomiting, diarrhea. These are just very mild symptoms that occur within 24 hours after the ingestion. But in a more severe cases, neurological symptoms such as confusion, disorientation, memory loss, seizures, and this can be detected within the 24 hours. So one of the prominent feature is this permanent short-term memory loss after ingesting this domoic acid contaminated shellfish. So it is like a hallmark of this DA intoxication. So we have many work done chasing this toxic species that producing the domoic acid, pseudonyxia. So over a decade of looking at our waters, what we have been documented not less than 30 species of this pseudonyxia with 9 new species discovery. I would like to highlight here is our current findings of a bloom of this Pseudonyxia cuspidata that occur in Miri that actually contaminated our shellfish. This is the rock oyster. So in another species that we often found it in our mangrove coastal water, we can see that the domoic acid also is a very common components or compounds that produce with this domoic acid detected as the major congeners in most of the strains that we establish. So as we know that local consumption on the mangrove and estuarine shellfish bivalve like clams and snails is very high in our country, we took an effort to sample randomly this shellfish from our local market and then screen for any potential toxicity. Indeed, we detected a DA, but with a very low level of DA in the flesh. But however, this has posed a health threat as not only short-term, but because the long-term DA toxicity may be associated with this kind of chronic exposure in people who are heavy consumers of bivalve. Okay, so now let's move on to another neurotoxin. This is ciguatoxin. There are more than 30 analogs reported so far. So this compound also considered one of the most toxic neurotoxin that we have. It binds to the voltage-gated sodium channels, and it somehow like excites the sodium channels and cause influx of these ions that triggers the action potential in the cell and leading to the poisoning syndromes. So what types of syndromes we have for this shellfish, for this poisoning? Again, you have gastrointestinal, cardiovascular, neurological symptoms, and this can occur within 1 to 6 hours after the ingestion of the toxic fish. The most indicative symptoms that we have for this kind of poisoning is this what we call as cold allodynia, like the dry eye sensation. You feel like it's characterized by burning pains when you respond to a cold stimulus. So this chart here show you how the different symptoms develop over time. So people may suffer this. It can last for months and years. So this aftermath is there. So this is something also unique for this poisoning. So in December 2017, there was a Ciguatera case that we managed to document the epidemiology, as one of the victims is a medical doctor. She actually provides us with a very detailed symptomatic description related to the poisoning. And we also took the fish specimens that left over and confirmed the toxicity by LC-MS. So following that, several cases has been reported in Sabah. Here you see is how we study the culprits, Gamberdiscus, that the one that produced this ciguatoxin. So these algae live attached to the bottom substrates such as coral rubbles, the seaweeds and sands. And to capture them, we will have to dive in and look for them, and just like playing hide and seek, but eventually we also managed to get them, and we confirm some of the species that found in Malaysia, in fact, they are toxin. You can take a look on our papers featuring this if you are interested. So to understand how reef microhabitats play an important role in structuring all these communities, the harmful algae communities, and to assess the risk of Ciguatera, we have conducted some ecological and physiological study on these species. And to put it simply, our findings pointed to the scenario that disturbance to the coral reef, that outer benthic substrate type, can have profound effect on our harmful algae composition, and it causes proliferation of these algae. So it is anticipated that if the coral reef is degraded, perhaps the species become abundant. This will alter the trophic transfer, and we will get more toxic fishes in our plates. Okay, we now move on to the last toxins that I'm going to introduce to you, pelytoxin. So this is again a groups of compounds that share a complex polyketide structure, you see here. So this is considered one of the most toxic non-protein natural compounds. So it was first found in zoanthid corals, and now we know that they also produced by some of the algae in the genus Ostropsis. So this toxin bind to another different way. The mechanism of action is different. They bind to this sodium potassium ATPase pump, and it causes them to become non-selective, and at a stage that they will cause severe cell and tissue damage. So toxicity of pelytoxin in human is not only through eating the contaminated seafood, but it may occur after physical contact with contaminated water, like you go swimming or bathing, or inhale the marine aerosol that containing this pelytoxin. So when it happens in the beach, then this certainly will affect our tourism. So and the consequences of this toxicity is also life-threatening. So this is the group of algae that produce the compound. Several cases of respiratory poisoning, skin or eye injuries like the dermatitis have been reported in beachgoers in the Mediterranean countries due to the toxic aerosol that released during the massive bloom of this particular species. So from the strains that isolated from Malaysian water, we examined the toxin production by LC-MS, MS, and we found that some of them really produce these forms of pelytoxin analog that we call ovatoxin and ostropsin. So it is fortunate that the density in our reef has not reached the level to cause aerosol poisoning. However, with the co-occurrence of all these potentially toxic genetics entities, it is worthwhile to further investigate the bloom dynamics of this species and its regulating factors before it's too late to do anything. So I've now come to the end of my talk. Well, I would like to talk a little bit about the challenges that we face in HAB monitoring. So to safeguard public health in terms of seafood safety and to have a better informed monitoring system is needed. But it is not easy. to have a perfect monitoring system, for many countries, this is somehow, we see the limitation there because there are baseline data that is not sufficient. We know little of when or where HARPs would occur, and often we only notice the occurrence of HARP species when they cause problems. So to have a better monitoring system, we will have to endeavors enormous work and efforts to samples, identify and quantify the species and the toxins. So to end, this is one of my favorite underwater photos taken from Perhentian Island. See who are they? The angry marlin and Nemo, and I envisage that this is the message to us. So with that, thank you very much.
Thank you, Dr. Liao, for sharing your knowledge and experience with us. So now we will move on to the Q&A session. Again, if you have any questions, feel free to type them in the chat box or raise your hand when we can unmute you. I think while we wait for them to type in their questions, I have a few questions collected. These are questions directed to Dr. Wong. So, Dr. Wong, someone asked you why you would like to go to the Antarctic to collect the algae samples.
You mean, so why? The question is why?
Yeah, why?
Okay. All right. Actually, this I have involved in this Antarctic research when I was doing my PhD, my postgrad study in 20 years ago. Yeah, I was given an opportunity to go to Antarctic to collect a sample as well as to conduct some field experiments. So these algae, they're actually you can't really see them in the Antarctic, but when you collect a sample back to the lab, you can actually grow them. Of course, you need to give them the nutrients and the appropriate conditions. And this algae is very interesting from Antarctic because they are in extreme environments, okay? So they are actually a very sensitive indicator for any changes in the environment. Yeah, that's the reason why my interest is actually a lot of focus on the Antarctic algae.
Well, Jenna, I hope Dr. Wong has answered your question. So the next question, also directed to Dr. Wong, is how can one determine which species of algae are safe for human consumption?
Okay, very good questions. This actually you have to do a lot of studies about the selected algae. As I mentioned, actually not all algae is actually toxic, but of course you need to do a very proper studies on those algae and to see what toxic compound that can be produced by algae. Of course, one of the simple things you can run those toxicity testings by using the mammalian cells. Yeah.
Okay, so the next question is from Kazan. So the question is, he wants to ask about the role of algae in bioremediation. Is it only enroll in nitrogen fixation or other mechanisms such as uptake of contaminants and so on?
Yes, algae is able to do the bioremediations. Actually, they have a few mechanisms. For example, they can degrade some of the toxicants by using some of the metabolism. Of course, it will involve the enzymes. Okay, some of the pollutants that will actually absorb the bioabsorptions at the cell wall of the algae, and that's how the algae manage to remove the toxicant from the environment.
Okay, so the next question is that if we use algae as a biofilter in, let's say, a shrimp pond or a fish pond, can it be consumed by humans or at least used as animal feed because it's accumulating contaminants?
Yeah, I think I have answered this question in the chat box, but let me explain again. Pollutants is actually, it can be bioaccumulate and also biomanufactured in the food chains. So normally for those algae that we use for bioremediations, we will not use it as a consumption as animal feeds or the human feeds, because it do contain the toxicants, unless we remove the toxicant from the algae. Okay, but they will be costly. So normally those biomass after the bioremediation, the algae biomass after the bioremediations, we will use it as a feedstock for biofuel productions. Yeah.
Okay, I think we have a few kids in the chat box who are really active with their questions. So they're asking like, if a fish can eat algae with color, will it change the color? Very cute, huh? So I think kids, if you are very interested, I think you can Google most of the questions, and we will continue with questions that we've collected for directed to Dr. Liao. So Dr. Liao, some
Yes.
is interested to know the occurrence of HAB in, I think it's harmful algae bloom in Malaysia over the range of a few years. So if this person wants to know more about such occurrences, so do you have any suggestions or recommendations for him to find other sources, maybe any papers that you would recommend him to read, anything like that?
Okay, we have quite a number of research papers working on harmful algal blooms in our waters. So we have 2 review papers talking mainly on what happens in Malaysia and also in the Southeast Asia. So I'm not sure who is in this, who is giving out this question. Perhaps later I can drop in some references in the chat box. Is it appropriate? Or because I can't really have these questions answered, because it's just some of the recommendations. So I guess, or you can contact us personally, then we can somehow email the information.
Sure.
Yeah, right.
So Jeremy, I think you can message Dr. Liao in the chat box, direct message her, or maybe she can drop some references in the chat box so we can all see it together. So the next question is like, what is the main indicator we can say that the bloom has happened?
Okay, this is a very tough questions, I would say. So when we talk about blooms of algae, so when you think of it simply, they are just like plants. What they need is just light and nutrients and sufficient carbon dioxide. So these are things that will triggers their growth. They will be very happy. They will grow very well when they have all these conditions favorable. So, but of course there are different. There are a lot of factors that will affect which species to bloom. So, like some species, they may outcompete the others because they prefer some kind of nutrients that is not favorable by others, just like what I mentioned here previously, ammonia. So ammonia mainly will be toxic to many algae, but some species, they can utilize this ammonia and thrive in the environment. So then they will bloom. So if let's say this particular species is the harmful one that they produce toxin, then it will cause this what we call harmful algal bloom. So it's very species selective. You don't find the same species everywhere. So somehow there are some kind of triggering factors to cause this kind of succession from one species to the other. So different scenario for different species, different location. Yeah. I hope I answer the questions. So there is no very concrete kind of indicators to say that, okay, we have this, then we will have bloom. No, it's not that straightforward.
Well, I think I hope Dr. Liao has answered your question. So the next question is that is there any competition among algae in the region?
Yes, yes, certainly yes. So that there are these allelopathic kind of interactions. So we think that some of the chemicals or compounds that produced by algae may be toxic to the other species as well. So they compete over space, compete over nutrients in order to grow. So these kind of competitions happen. So you will see a succession from one species to the other when the environment change. Yes.
Okay, so I think the below questions are I think open to both doctors. Hold on. So I think someone asked if it's possible to use the toxins produced by the microalgae as anti-cancer.
There are research that is ongoing. So we don't have, there are preclinical kind of studies working on all these compounds. We know that some of the compounds that I mentioned previously, they have been used in like for local anesthesia because they work on our neuron. So with a moderate kind of concentration, it can be used as a local anesthesia. And saxitoxin, one of the analogs that actually used as a treatment for Botox overdose. So that is some of the works that have been done on the pharmaceuticals point of view. So for those that working on cell lines like anti-cancers, I think that there is not much studies that had actually come out with a very concrete evidence yet. But as a local anesthesia, that is something that is somehow is already in a post-clinical stage. Yeah.
Right. So I just have a last few questions before we end. So one of it is how to overcome blue-green algae bloom in earthen culture pond, because the blue-green algae will affect the water quality and cultured shrimps or the fish. So do you have any advice to deal with this kind of situation?
It is, I think, should I answer this or Dr. Wong would like to have this answer?
I think Dr. Liao, you can go ahead.
Okay. It's more or less the same as what we see from the marine environment. So when we are talking about the earthen pond, especially these artificial ponds that we use for shrimp farms, the water quality is the one that we should monitor. So blue-green algae is common components in the freshwater system. So if we don't control the water well, like if you have too much of nutrients added in, and then the species will grow, they will thrive. So the water quality is the one that we should monitor to ensure you have what you want. Because in most of these kind of aquaculture ponds, we want to have some good algae there, but we don't want some bad algae. So you have to play around with some environmental conditions, the water quality, in order to control what to grow in your pond. Yeah.
Okay, the next the last three questions I think are directed to Dr. Wong. So Dr. Wong, do all types of microalgae contain the same type of organic compound, such as anti-cancer, antibacterial, and so on? Or is it the amount that differentiates them?
Different algae, because they are, I think, more than 50,000 of microalgae already been identified. And all of the algae, they are actually, they have different characteristics. And of course, in terms of the bioactive compound, different algae, they were producing different bioactive compound. Yeah.
So, what are the challenges of microalgae cultivation in the laboratory?
I would say there's a manpower. Okay. We actually similar in my lab. Actually, I'm also facing the problem of manpower to maintain the culture, the algae culture, because it's really time consuming. It's the job is actually quite routine, but just that we need to make sure the algae is they are free from contaminant, especially the cross contaminations. So and we need to routinely to grow the culture, maybe once a month, to maintain the freshness of the algae. So the manpower, I think, is my bigger challenges.
So, are there any differences in the treatment of microalgae cultivation in the laboratory for bioenergy and bioremediation research purpose?
Again, I can't get you, Yongjin.
Someone asked if there are any differences in the treatment of microalgae cultivation in the laboratory for bioenergy and bioremediation research purpose.
Different treatments. Actually, I can't get what you means by the different treatments.
Actually, this is the question collected from the chat box. So maybe this participant would like to unmute yourself and ask Dr. Liao, Dr. Wong, personally.
Yes, hello. What I would like to know is about how you deal with microalgae cultivations. Is there any differentiations in dealing with the cultivations for bioenergy and bioremediations research purpose?
I guess in general, what is important to get the algae products from the algae biomass is we have to make sure the biomass from the algae is enough for us to produce the biomass to produce those compounds that you want, including the variable biofuel or bioenergy. So it's very important that the how do I put it? The culture system must be very good. Like for example, you have to give the algae sufficient light. Of course, if you give if the light is over intense in intensities, then it will the algae will actually facing the photostress. We will actually kill the algae as well. So a lot of adjustment or optimum condition, I would say that is very important to make sure that the algae grow rapidly and grow healthily. So I think one of the methods to grow these algae into mass scale is actually bioreactor. Yeah, because bioreactor, it can actually it's a closed system whereby it can protect the algae from any other contamination. But of course, the cost will be very expensive compared to other methods. Yep. I hope I'm answer your questions.
Yeah, thank you, Dr. Wong.
Thanks.
Are there any more participants that would like to unmute yourself and ask something? Perhaps I see these three students that are very active: Emirna, Itessa, and Idelphia. Maybe you would like to unmute yourself and say something. No? Well, thank you for everyone's enthusiasm. So before we proceed with the quiz session, we have a photo session. So everyone turn on your cameras and we will capture this moment. Again, you can switch to the virtual background we have provided. And photographer, when you are ready, please let me know. Right, I will count on 3, 2, 1. Okay, one more. 3, 2, 1. Okay, the last one. 3, 2, 1.
Okay, all good.
All right, thank you, Shinman. So now it's time for our quiz session where you can test your knowledge on the curious chemistry of algae. Our quiz master today is Mrs. Mandy Sum and Mrs. Jacqueline Fu from Access Dunia, who will be asking a series of questions related to our team. So let me explain a little bit about how it works. They will ask a question, and participants, you may type your answers in the chat box. The first participant with the correct answer will win a 50 ringgit Grab food voucher. So make sure you pay attention and be quick on the draw. But that's not all. We also have a lucky draw at the end of the quiz session where 5 lucky participants will win a special prize. So stay tuned and keep your fingers crossed. So without further ado, let's get started. Over to you, Mrs. Mandy.
Thank you very much. Thank you. Can you hear me?
Yes.
Thanks. This year is a very special year because due to the topic this year, algae is a very common thing. I see our participants come from academic, some students, I think, and also from some of the specialists in the field. Yeah, and that will be a good sign. For the Earth Day is more toward the awareness. Yeah, awareness for everyone's to aware the unique creatures in the world. So this year, when we celebrate the Earth Day, is the actual day 22nd April, but it's due to the holiday, so we make it a little bit earlier. And this week, we celebrate the Chemist celebrate the Earth Week, and the theme is the curious chemistry for the amazing algae. And thank you for Dr. Wong and also Dr. Liu sharing what will be the understanding about the algae. I think a lot of questions been asked. When I look into all these questions, okay, I found that this is a lot of creative thinking, the thought coming in, which I think when the longer we are in the workplace or in the academic world, we are start to like forget the curiosity really make a difference in our work. Yeah. So for our pre-session, it's very simple. Okay. To celebrate the Earth Day, we give a small little token of give to whoever want to participate. My question is very simple. We are not testing the technicals or any of the special knowledge, but it's give you opportunity, okay, to have a chance to give a little prize, okay, during this week. Okay, whoever is the faster person who give us the answer in the chat box, my colleague Jack, she will help me to spot what will be the questions and the answers. The first one who answer will have the prize, and we'll give a chance to everyone. So whoever win it, so the second time we'll choose the second one to win. Type your answer into the chat box, and we will have the right to decide who gives the correct answers and who is the winner. Okay, and the winner, please stay back at the end of the session so we'll be able to identify your name and the detail in order to send the prize to you. And the prize is each questions we give the Grab food voucher. Yeah, so this is a 50 ringgit equivalent. So because we open up the Zoom for the Southeast Asia countries, so this will be based on the Malaysia ringgit 50 equivalent amount. Okay, are you all set, ready? Okay, a very simple question to ask. The first question is, algae have evolved, produce and store energy, and more than what percentage of oxygen in our atmosphere come from algae? Can you all type in your, okay, very fast. Even before I finish the questions, you already have the answers. Jack, do you manage to identify the winner?
Yes, it is.
I think the winner, the first one is Etisa. Yeah,
Yes.
Etisa, give us your email address and the contact so that we'll be able to reach out to you. Okay, I just want to debrief for this. It's very not technical. It's almost half of our atmospheres. Okay, oxygens come from algae. So this is a very, very useful that we want to preserve our ecosystem. So algae is a really amazing creatures. Okay, it produces the whole ecosystems with the food, with some of the special elements. that really help us to balance the ecosystem. Okay, my next question is, okay, very simple. If you attend the sessions, you'll be able to answer. There are basically how many types of algae based on their color? Okay, so we find the answer, but make sure we provide to the person who first answer. And Jack, do you manage to identify the winner?
Yes, it's injured saffron.
Oh, okay, inject saffron. So we open up to all the committee members to participate into it. There are basically 4 types of algae. Yeah, the answer is 4. Okay, based on their color: green, blue-green, red, and brown. I think if you attend both our speaker session, Dr. Wong and Dr. Liu did mention a little bit, yeah, which category belong to the green, yeah, what example for the blue-green, the red color, and the brown color. So not technical, everyone can answer. Hope you can stay put for this. And the next question is, what is the color of the light in the ocean? Well, meaning of this tiny organism called dinoflagellates that live in the water. Okay. Jack, do you manage to find the answer? Make sure it's not the same winner.
Yeah,
If the same
Mr.
winner, we give the
Ivan
chance.
Sam.
Okay, thank you. So this is a very simple, I think Dr. Wong and also Dr. Liu did mention. Okay, that will be a type of a single cell algae called dinoflagellate. And this tiny creature will emit a small amount of the light. Yeah, this will be make the oceans very colorful, because that will be the whole process, okay, that we're going through to differentiate. Okay, the answer is a blue. Let me keep, okay, the answer is a blue. So the next one even, okay, and why is a blue color? Yeah, because there will be a chemical section called luciferic. So this is responsible for the glowing. Okay, it's very similar into the green chlorophyll in the leaf of the plant. Okay, when they react with the oxygen, they produce a blue color light in the ocean. So this is an amazing creatures there that will make a wonderful wonder in our ecosystem. Okay, the question 4. Macroalgae and the macroalgae contain oils that can be used as what? And it's biodegradable even if it's accidental spills. It does not hurt the environment. What's the answer of it? Okay, Jack, do you manage to find the answer?
Actually, okay, they are not
Biofuel can be accepted.
can be accepted. Okay.
Yeah. Can you please identify the
sorry.
person who typed the biofuel?
So it's no Shahida.
Yeah. Actually, my answer is very simple. Biofuel is like equivalent to it. Yeah. So this will be around the world. A lot of scientists looking for ways how to make our energies, okay, not only depend on the one source. So biofuel, okay, including from the algae and other living things, that will be one amazing possibility that we can look into. Yeah, because that will be able to produce a certain oils and can be used at the biofuels. Yeah, so this will be something that we look forward for the human. We are living into our Mother Earth. We need to take care of it. So if it's something we can do to improve it, that will be the responsibility for everyone. Okay, my last questions. I think whoever attends Dr. Liu's session, you will hear a lot of this. Algae grow normally in the United States in the coastal water, but when they grow, get out of control, what is called, acronym can be accepted. So Jack, do you manage to identify who is the winner?
Yeah, soon.
Okay, the answer is harmful algae bloom. Okay, or we call it HAB. I think it's been mentioned a lot by Dr. Liu's actions. Yeah, what will be the activity? And it happened in Malaysia as well, and in our the regions. Yeah, this will be the excess nitrogen and the phosphorus cause an overgrowth of the algae in a short period of time. Yeah, so this is called the algae bloom. And this will be the one we use a lot of oxygen and block the sunlight from the underwater plants. So eventually that our microorganism under the water itself, we won't be able to get oxygen and also the sunlight. So that will be a great harmful thing to us. Okay, I just would like to thank everyone who participated into today's sessions, and we're going to have a lucky draw. I will pass it back to the organizer. Yeah, thank you everyone to participate into this session. Hope you have a great memory for today's sessions. I pass it to our organizer.
Yeah, thank you, Mrs. Wendy. I hope everyone had fun during the quiz session. So now we will move on to the lucky draw. So there will be a spinning wheel where we will pick 5 lucky participants, and you will win some limited edition merchandises from ACS, and also a special collagen drink from Boom. So without further ado, multimedia team, please present. All right. So this is a list of all the names that are joining us, that are with us today. And now we will have our first lucky winner. Okay, so congratulations to Wong Chun Yin for being the first winner. Please put your info, your email address in the chat box so that our committee can reach out to you for the prizes. Please, let's move on to our next winner. Okay, so we have a second winner. The second winner is Dan Chow. Remember to put your info in the chat box as well. Now for the next one. So congratulations to Jaffa La Kilo. Now our fourth winner is Farida Khalid. Okay. I'm sorry, Fazira Khalid. Okay. And our last winner. So our last winner is Nafal Nazar Bajat. Congratulations to all 5 lucky participants. Please remember to put your info in the chat box, yeah? Okay, so now now that we've come to the end of our session today, so I just want to thank a few people here. So firstly, thank you, Dr. Wong and Dr. Liao, for your expertise, and also Dr. Lim Po Tin for helping us with the questions in the chat box. Next, I want to thank Mrs. Mandy Sum and Mrs. Jacqueline Fu from ACS Publications for generous support, and also our ACS advisors, Dr. Vanajan, Dr. Woi Pei Meng, and Dr. Amirullah. Next, I want to thank our main sponsor, Boom, and ACS Malaysia for the prizes, and also all the committee for your efforts. Now, before we leave, I would like to take the opportunity to promote our next events. So the first event is World Environment Day 2023. Our theme would be Solutions to Plastics Pollution. It's happening on the 5th of June at 9 o'clock to 11 o'clock, and we have 2 speakers here, Dr. Imenda Sembiring and Dr. Jane Giulaiti. So be sure to scan the QR code so you can join us on our next event. So this is an online event, and our physical event will be Algae in Wonderland, happening on 5th of May on Friday. It will be at 11 o'clock to 1 o'clock at the Student Lounge, Level 1, Bangunan Magma Kimia UM. So this is open to everyone who wants to join, as long as you can attend physically on that day. And we have a registration fee of 5 ringgit per pax. So we are going to have hands-on experiments, station games and booths, and also foods and prizes. So make sure you join us, yeah? That is all for the promotion. So thank you everyone for attending our event. We hope you enjoyed and learned something new today. So we'll see you at our next event. Bye-bye. Oh, right. Sorry. Before you leave, please make sure to scan the QR code and give us some feedback, because this we will send out the certificate upon the completion of the feedback form. So make sure everyone scans the feedback form and make sure you type in your name so we can send your certificate to you. Thank you. So I would like to remind all the winners just now to share your email in the chat box or by a private message if you haven't done so, okay?
Thank you.
Can we do a quick interview to these 3 active participants in our chat box, Edessa?
Sure.
Yeah. Where you get our contact? Is someone give you our link? How you get our events? Dessa? If you can unmute yourself, then we can have a chat, because I found this is the first time we encounter, okay, a lot of questions asked, yeah, from our 3 young audience. I would like to know what will be your plans after you're here and join our event today. Dessa? And another one I see missing.
Adelphia,
How
she
to-
gone already.
Ah,
She's
Dessa?
gone already. Yeah?
Are you siblings? You all look alike.
Yes, we are siblings. All 3 of us.
Ah, so I see you have a lot of curious questions.
Adelphia, she's
Whether
looking for
this,
you.
yeah, we can do a quick interview, because I stopped
can't hear.
recording and all this. So whoever stay back, yeah, we'd like to ask this 3 audience, yeah, being the young generations. Hi.
Hello.
Yeah, I see you type a lot of questions. Can you ask personally?
Do you have more questions to ask? No. I
Yeah,
also
you
don't
ask
have,
whether
I don't have questions to ask.
Algae whether got the COVID or not.
Oh, that's a Myrna.
Where you get our link Zoom from? Is your parents?
Mommy.
Who is your mommy?
What's your yi?
Again?
What's your yi?